Okay, great. So today I'm going to talk about de novo protein design, which is building proteins, brand new proteins from scratch um, without using um, native protein, not redesigning native protein structures, so building brand new proteins from scratch. And for many years, we have been doing this using a physically based model of folding embodied in the Rosetta software. And here, the basic concept is that proteins fold to their lowest free energy states. So if you want to design a sequence which folds up to a structure that you're interested in, you have to find, well, if you have a structure, if you want to make a new structure, you first come up with that structure, and then you have to find a sequence whose lowest energy state is that structure. And so to do this, you have to be able to calculate the energies of different conformations of a protein chain with different sequences. And you have to be able to sample in sequence space and in structure space. And so in Rosetta is software that does all these things. Now, as you all know, deep learning has become very much more powerful, particularly when applied to computational structural biology. And um, in and we've been using deep learning extensively for protein design. And here, the basic idea is that if you have a network which maps sequence to structure, then if you want to make a new structure, you have to find a sequence which the network predicts will fold to that structure. And here, it's not based. It's not based on sort of an energy, an energy function. We're not calculating hydrogen bonds or van der Waals interactions as in Rosetta. Here we're simply taking the network as a mapping from sequence to structure. So in the first part of my talk, I'll talk primarily about work we've done with using this energy-based picture of folding, and then towards the end, I'll focus on sort of what we're doing now with deep learning approaches. And it's actually interesting. As far as what we're doing in the lab, it really hasn't changed. Just what's going on, what the software at the very bottom is getting sort of like the energy-based methods are getting augmented by the deep learning methods. But I wanted to begin. It's a very exciting time for protein design because on July 29th, the first de novo designed medicine was approved. This is a COVID vaccine based on self-assembling nanoparticles that if you had heard me, if you heard me talk maybe 5 or 6 years ago, I would have described the methods for designing self-assembling protein systems like icosahedra from 2 different protein components. And I actually look back, we published the paper in 2016, and on June 29th, so my colleague Neil King has been putting viral antigens on these nanoparticles, and they elicit very strong immune responses. And so the phase 3 trial looked great, and people are now getting this vaccine, and so it's very exciting. So I'm going to show you a lot of crazy stuff, but maybe it'll become useful. And it only took 6 years in this case, so which is pretty cool. All right, so also on the coronavirus theme, and what I'm going to talk about at the beginning, this first section of my talk, is the design of binding proteins. So here the problem is you're given an arbitrary protein target, and there's a particular site on the target, and you want to design a protein which binds to it. So basically, we generate large numbers of protein scaffolds in silico, and then we dock them against the target and design the interface for shape complementarity and complementarity and chemical complementarity. And then we make the proteins and test them. Here are what some of these proteins look like. So this one is binding at a site that's this one. Well, okay, let's just say this is what they typically look like. This is a 3-helix bundle. It's just 55 amino acids. It's stable at greater than 95 degrees, and And when the cryo-EM structure was solved in David Wiesler's lab, it was found to be nearly identical to the computational design model. Here you see 3 of these proteins bound to a single spike, and this is a superposition of the cryo-EM structure and the computational model. So this protein binds very tightly to the spike, and it neutralizes the wild-type virus very effectively in both cell culture and in animals. But we wanted something even tighter, so we took advantage of the fact that these proteins are small, so 3 different copies can bind to the same spike. And so here we have we've designed a trimeric protein, trimeric version. So this is a de novo design trimer that holds 3 of these binding modules in the right relative positions to interact with 3 RBDs all at the same time. And this protein is very potent. It neutralizes Omicron and all the recent variants very potently, both in cell culture and animals. And this is not as far along as the COVID vaccine, but it will be in clinical trials next year. So we've used this approach to make binders to a wide variety of cell surface receptors in addition to viruses, and some of them are listed here. And we're very interested in these because what we can do is array them in different arrangements, and this shows an experiment with the FGF receptor where we've arrayed 6 of these along on the outside of this designed hexamer. And in this case, what this is basically we add these to cells, and the cells respond very strongly to FGF, and they also respond very strongly to this completely synthetic agonist. But it's different. It's interesting because depending on how you construct this agonist, this how you array these FGF receptor binders, you get very different effects, and that's very interesting. And we have all these different binding domains now, and we can arrange them in different ways. And we're trying to explore the range of signaling activities we get. Another strategy for so this design strategy I described before sort of relied on having a structure, but we can also design binders for peptides that are disordered. So in this case, this work from Danny Sato. I should say the developers of the protein design method are Longjing Cao and Brian Coventry, and many people were involved in all the work leading up to the pointing towards the clinical trials, and I'll acknowledge them at the end. But in this case, so this work by Danny Sato, and he's designing proteins which have a slot in the beta sheet where an exogenous peptide can slot in, and these peptides are interacting through both hydrogen bonding on both sides, as well as the hydrophobic residues that are sticking in the protein. And these proteins bind to their targets very tightly, and Danny has extended this to make specific binders for the abeta peptide, and he sent these to his collaborators at Tuomas Knowles Lab, and they have a very nice assay for amyloid formation. And so basically, if you take the abeta peptide and you put it in solution, it very rapidly aggregates. That's the gray line. But when these binders are added, they at stoichiometric concentrations, they completely shut down amyloid formation. And these are, in fact, according to Tuomas, the most potent anti-suppressors of amyloid formation that they've seen. So we're very excited for the potential for protein design now helping with amyloid diseases. So far, those proteins I've described are all static, but we can also design systems that undergo conformational change. So here we have a designed cage, which cages a binding domain that is high affinity for a target. So when the target is present, that causes the whole thing to open up. And when it opens up, this key can bind the cage, and that reconstitutes luciferase activity. So we get light emitted in the presence of the target of the binding domain. So as you can see here, what's being illustrated is this concept in the case where the carbon brothers binding domain I described is inserted into the sensor. And this works very well. We get rapid increases in the amount of light emitted when, and there's no mixing required. This is just all going on in solution. And we're using this now to make sensors for a wide variety of different compounds of interest. We can use a similar concept to do calculations on the surface of cells. So here is, so if we have a cancer cell that has two markers that are individually found on healthy cells, we want some way of targeting only the cells that have both markers. And so we can target two components of a switchable system to the two separate markers. And then only if they're both present do we get exposure of an effector region. In the case I'm going to show you, this effector region is recruiting a CAR T cell. And that's shown here. So here we have, this is actually a little bit more complicated. We also have a healthy cell marker, and we want the signal, the recruitment, to get shut down in the presence of the healthy cell marker. And so you can see here we're only getting a recruitment of the CAR T cells and activation of them on cells that have the two tumor cell markers, but not the healthy cell marker. And cells that just have one of the markers get no activation at all. So we can do, in this case, this is A and B, not C logic on the surface of cells. So kind of this is more general direction we're excited about, being able to make medicines that are more precise than current ones. I wanted to show you that we can, that one can use protein design outside of the medical realm. And so this is a, Nate Ennis has been very interested in the photosystem, and at the heart of the photosystem is what's called a special pair, two chlorophyll molecules that are held in just the right arrangement for for energy transfer. They're exotonically coupled, and so he's designed. But photosystem is a big, complicated protein. He's designed synthetic dimeric proteins that hold the two chlorophyll molecules in exactly the same position they are in the photosystem, and this compares a crystal structure of this design model to the arrangement in the photosystem. And what's really cool now, is a very good model to study, and we're working with spectroscopists really around the world to characterize the photophysics of this. And there's clear excitonic coupling between chlorophylls. Well, we can go further. He and Chenji Wang used the methods I alluded to at the beginning to for designing these self-assembling systems to design self-assembling cubes, as you see here, where each edge of the cube is one of those chlorophyll dimers. And so we now have the starting point of a synthetic light harvesting system where with two chlorophyll molecules on each edge of this cubic structure. So I've now described several different self-assembling systems, and we want to dive into one in a little bit more detail. So we've designed proteins like these green ones here, which bind to antibodies through the Fc region and cause the antibodies to assemble into icosahedral structures. And we have other ones like this tetramer here, which hold the antibodies in just the right arrangement to wrap into an octahedral structure. And we've solved cryo-EM structures of each of these, and you see there's some other ones here like tetrahedral structures. We found a number of interesting things with these. First of all, you can add almost any antibody, and it will add to the corresponding green protein here, and it will assemble it into a nanostructure. But these sort of antibody assemblies have greatly potentiate cell signaling because they cluster the receptors. What I want to focus on today is that we're very excited about these for delivery. So the basic idea is we have the two components, we mix them together, and this is why I just showed you, we get these octahedral nanoparticles. They have an open threefold axis, so if you put cargo in, it would just leak out. But Aaron Young, a grad student in the lab, has designed trimeric plugs, which fill that open space And uh, and here's the cryo-EM structure of looking down the four-fold axis on the two-fold symmetry axes. If we look down the three-fold symmetry axis where the plug is. But the cool thing is that Erin has designed these to be pH dependent and actually quite pH dependent. So the plug will actually come out at around, these are different variants she's made, but even at pH 6, we have variants that almost completely come apart. And that's interesting because in the endosome, the pH drops. So the idea is we can put a therapeutic RNA, like a guide RNA, inside and assemble these with antibodies that target the cells or tissues we're interested in. They'll get taken up by the endosome, and then the RNA will get released for gene editing or something else. And we have some preliminary results with editing already using systems like this from Isa Yosef, another grad student in the lab. So I've described the assembly of these closed nanoparticles, but we can also design now, Zhe Li and Shanji and Una have figured out how to design extensive protein crystals. So of course, there's many, many protein crystal structures that have been solved, but it's been very, very difficult to actually design crystal structures. So basically build an atomic model of a lattice and then design it. And so here, the strategy was hierarchical to first build up cages, nanocages like the ones I showed you, that have surfaces that can interact well with each other, like here, and then to design those interfaces. And I'm showing you pictures of the crystals that form. And then also these crystal structures are very close to the design crystal lattices when we solve their crystal structures. One of the things that sort of was sort of shocked structural biologists like Max Perutz when the first structures were formed, is that proteins were so irregular and so asymmetric and so non-modular in contrast to DNA. But now with protein design, Tim Huddy has figured out, and his colleagues in the lab have figured out how to make proteins that really are more like the building materials we're used to thinking about outside the protein realm. So you can make perfectly straight proteins, and you can shrink or contract them just by adding or taking away repeats. And so he's made these protein squares as shown here. And to make bigger squares, he just extends the length of the building block. And here you can see the EM structure of the bigger squares. So we get scalable materials without really having to do any redesign. We just expand or contract the building blocks. Here is you can see the image here. Here he's got one of these straight repeat proteins here and a dimer here. And he calls these train tracks for obvious reasons. And again, I'm not showing it here, but these train tracks are expandable. He can bring the two tracks closer together and change the spacing of the struts. So it's kind of opening the door to much more modular design of nanostructures. We can also now design cages from these and change their size just by varying the number of repeats in these arms. And an example is shown here. So a final example I'm going to give you of designs made using sort of these traditional Rosetta methods is the design of, or steps towards the design of rotary machines. And this is work from Alex E. Corbet. So here he's designed axles and rings and designed interfaces between the axles and the rings. And he's worked out ways of assembling the rings around the axles. And so what that has enabled him to do is to design a wide variety of systems with rings surrounded by different types of rotors. Axles surrounded by rotors. And in some of them, these are now cryo-EM data here. In some of them, the rotor does not adopt a well-defined structure relative to the axle. And if you look at the energy landscape, the rotational energy landscape, you can see it's pretty flat. And this happens often when the symmetry of the axle is different from the symmetry of the rotor. And this actually, you could argue, is good behavior for a rotary machine because it's not deeply trapped in one state. But it's a little bit hard to structurally characterize. So this system, on the other hand, is interesting, and I'll show you it. This is a C4 axle, D8 axle, and a C4 rotor. And in this case, there are two rotors that assemble onto the axle. And with EM classification analysis, Alexey can distinguish two different rotational states of the system, one in which the rotors are staggered and one in which they're eclipsed. So now we're trying to use single molecule measurements to follow the rotation of the axle, of the rotor around the axle. Okay, so now I'm going to switch over to the deep learning part of my talk. So after we learned about the exciting advances of DeepMind in the last CASP, we of course were kind of intrigued, and it wasn't really clear at that point whether DeepMind was going to release the code or describe in detail how they achieved the very high accuracy. So we sort of built off some of the concepts they introduced at their talk, and we developed roseTTAfold. We've now extended that. This will be releasing a new version very soon, and one of the things that's different about roseTTAfold and AlphaFold is it has, so AlphaFold, of course, has sort of a 1D track and a 2D track. roseTTAfold has a 3D track with coordinates that sort of go in from the very beginning, and so there's a coordinate representation. And for experts, one of the things that's nice about that is you don't need, one of the things that takes a lot of memory in AlphaFold is triangle attention, because you have to, if you want residues that are far apart from each other to attend to each other, you have to basically have an attention map, which is very, very large. Here, since we have a model of the 3D structure, we know which residues are close in space, and we can pass that information in, can bias attention by having going from the 3D track to the 2D track. But this also, and more relevantly for my talk, makes a lot of design, is nice for design applications, because as I'll show you, we can feed in partial structures and complete them by going through the network. So this is roughly where, so the original roseTTAfold was clearly not as good as AlphaFold. And the new roseTTAfold is comparable to the better of AlphaFold, or AlphaFold multimer on monomers and complexes. So it's gotten a lot better than it was before. And we've learned a lot in getting from here to here. So now I want to show how we can use these methods for protein design. So the first idea that Ivan Anashanka came up with this is what we call network hallucination. For this, in the machine learning field, this would be called activation maximization. So we start with a random amino acid sequence, we feed it through the network, and the network is not, it's a random sequence, so it's not predicted to fold up to any particular structure. But then we can optimize the sequence to the point where it's predicted to fold to a well-defined structure. And what this looks like shown here at the beginning, there are not well-defined contacts. And then as we go through the sequence optimization, we get sequences that are predicted to fold to better and better defined structures. And so these are just 3 different trajectories, starting with different random number seeds, and you can see they lead to very different types of structures. And we have solved a number of these, and the structures are quite close to the design models. When we tried doing this for more complicated sorts of structures, we found that the design sequences, when we made the proteins in the lab, they often aggregated. And so luckily, we in parallel had been, and this is work from Justus Duparas, a postdoc in the lab, had developed a very fast ML-based method for protein sequence design that relies on a message passing neural network. And one of the nice things about this is that you can tie positions during the design, during the decoding process. And so you can impose different types of symmetries and also do multi-state design all within the same framework. And one of the things that was a bit of a shock to us is that this method, protein MPNN, is very much more successful in the classic problem of predicting taking a native protein backbone and predicting a sequence for it. And so with Rosetta, on average, we get something like 40% sequence recovery. And in fact, a lot of Rosetta over the years has been sort of optimized for this. Whereas with protein MPNN, we're up to 57%. And here you can see, this is at most buried regions, you can see they're pretty comparable. That's because it's a jigsaw puzzle. But as we go to more and more exposed positions, the protein MPNN can pick up on all these subtle features that Rosetta, as an energy-based model, has a much harder time doing. And the method works really well for, for different types of structures. So when we started doing hallucinations with AlphaFold, what I showed you before was with TR Rosetta, we made the proteins, they just didn't express. And so we made a bunch of proteins and we got essentially no soluble yield. But we took those backbones and redesigned them with the protein MPNN, and we got in most cases, but the proteins now suddenly expressed. The backbone is the hallucinated backbone, but the sequence is completely new, and the proteins are stable, and we've solved crystal structures, and they come out very, very close to the hallucinated models. So this has now enabled us to do more complicated things. So here, this is coming back to hallucination. We are not just hallucinating a single chain, but we're hallucinating multiple chains, and we're requiring cyclic symmetry. So in the loss function, in the optimization, we assess the extent to which the structure is symmetric. And this has led to the generation of a wide variety of new types of oligomeric structures. These are fairly complicated structures, as you can see here. Here's a very intertwined dimer, and we've solved crystal structures for 7 of them. And what I'm showing here is the superposition of the crystal structure on the hallucinated backbone. And so what's really cool about this is with this hallucination approach, you can explore really protein space in a very general way. And even though the networks are trained on native protein structures, there's nothing in the PDB that's very close to any of these. So you can, it's of course interpolating to some extent, but the protein structure database is so rich, what comes out is really quite new. So this has gotten to a point so advanced that over the winter quarter of this past, just earlier this year, 2 rotation students were assigned the task of, or the project of taking this to even more extreme level of, say in this case, going out to 33-fold cyclic symmetry. So they start with a short random repeat, and then they copy it over 33 times and generate structures and do this hallucination process. And what came out of this were these giant rings. Here are cryo-EM structures that Alexis Courbet solved. You can see they're really beautiful structures. There's nothing really in nature that looks like these. So basically, again, sort of we can explore protein space in a totally new way now. So now I'm going to switch over to using in the last few minutes using protein design to design new functions. So the first one I'm going to talk about is catalysis. And so Andy and Chris Norn have been interested in designing luciferases, and so they focused in on this rate limiting step in the luminescent breakdown of a synthetic substrate. And they designed, first Chris used the deep learning methods to generate large numbers of scaffolds, this hallucination method, to generate large numbers of scaffolds with pockets that are the right shape to hold the substrate. And then Andy designed a binding site for us to stabilize the transition state for the reaction as shown on the right. And it's really neat. What came out of this were designs that were already active, and with just 2 additional mutations, had kcat over kms on the order of 10 to the 6 per mole per second. And they're very bright. This is just looking at a tube, which tube of cells. And what's even neater is that the native luciferases have big expansive pockets, and so they don't really distinguish between different substrates. This just shows activity on a range of substrates. This is the substrate that Andy focused on. whereas this de novo luciferase is very specific for the intended substrate because it's got that really close complementary pocket. So we're excited about this now for being able to multiplex luciferases in the same way that people can multiplex fluorescent proteins. Okay, so in the final few minutes here, I want to show you how we can now constrain this generative modeling to make proteins with new functions. And we've been looking at a variety of different types of functions. For example, the design of enzymes, like I described, the design of proteins which bind to other proteins, and the design of proteins which scaffold antigenic epitopes. And so the first approach I'll tell you about is what we call constrained hallucination, which is like the unconstrained hallucination, but we take a binding motif and we add to the loss of that's guiding the sequence optimization, a term which evaluates how well this binding motif is recapitulated in the design structure. And so this now shows the use of this constrained hallucination to design proteins that scaffold an epitope or epitopes from the respiratory syncytial virus. And the designs are shown here. So the gray is the hallucinated part. The red and orange part is the part that we were keeping fixed. And you can see the hallucination has built proteins which scaffold these epitopes. And we've made these in the lab, and they bind the neutralizing antibody actually quite well. And they're sort of promising new candidates for respiratory syncytial virus vaccines. And here's the same concept now applied on ACE2 to build proteins which scaffold different parts of ACE2. We can use the same concept to design proteins which hold catalytic residues together in particular geometries, in this case specifying the side chain coordinates and then hallucinating around them. This shows you the design of a proto-carbanic anhydrase, and here we have a ketosteroid isomerase. So a perhaps more elegant approach to the same problem is using roseTTAfold to inpaint missing sequence and structure. So the hallucination approach is very nice because you can customize it by changing the loss to whatever you want, but you have to go through the network many, many times. So instead, what we can do is to train roseTTAfold not only to go from sequence to structure, but to go from to start with a known structure and go back to sequence. And then we can also train it. We can give it partial sequence and structure information as indicated here, and then have the network fill in the missing sequence and structure information. This is pretty much like viewing roseTTAfold as a language model, but not as a sequence language model, but as a model that understands the joint sequence structure language. So we train it by masking out parts of the sequence and structure and then ask the network to recover it. And this method is very nice because you give it partial information, and then in a single forward pass, it fills in the missing sequence and structure. And the sequences that come out from this are actually better than the hallucinated ones. So this is an example where we take metal binding sites and we expand out, we just feed those in, and then the network just builds out the rest of the structure. And here's, this is a di-iron site and an EF hand calcium binding site. And we've made the proteins, they bind metal. And so we're now using these approaches for a wide variety of function design problems. Okay, so yeah, thank you for your attention. And I just want to acknowledge the amazing people who've done this work. Longjing Cao and Brian Coventry developed the general protein design method. The development of the COVID therapeutic is the work of many people. Keija Wu designed the trimer, and Brett Case and Mike Diamond did the cell at WashU did the cell neutralization and in vivo experiments. Andrew Hunt and Mike Jewett did a lot of the testing of different constructs at Northwestern. Longjin and Brian and others in the group made all that long list of binders I showed you. Natasha and Thomas did the work on the FGF receptor signaling, the synthetic FGF, in collaboration with Yossi Schlesinger's lab and Honolulu Baker's lab. Danny Sato designed the amyloid binding peptide that could have therapeutic utility for amyloid diseases. Alfredo and Andy developed the sensor, and Mark and Jillian did the work on the logic for recognizing cancer cells and distinguishing them from normal cells. Nate Ennis did designed the synthetic light harvesting systems. Alexei designed the molecular machines. Oh, and I left out Tim Huddy, who designed the expandable materials. The protein hallucination work is done by, the original work was done by Yvonne Sam Temuka with structure determination in Guy's lab. Alexei, Elias, and Sam are the two rotation students who designed the giant rings, and Basil and Lucas designed developed the general method. The protein MPNN, which has pretty much completely displaced Rosetta for sequence design in my lab, is the work of Eustace Duparis. The denovo design luciferase is the work of Andy and Chris, and Drew and Sydney, Doug, and Sergey were involved in the constrained hallucination method, and David and Joe developed the inpainting method, and Ming-Kyung Bak is and Frank DeMaio have developed roseTTAfold, and Ming-Kyung has really been at the heart of a lot of what I've described. For example, the inpainting method directly builds off her work, and I've had many wonderful colleagues at the IPD. So yeah, I think I wanted to leave plenty of time for questions. So let's see, I'll stop sharing and would love to take them.
Let's thank David for this amazing talk. Yep, and we'll start with questions from the audience.
Hello, amazing talk. Thanks a lot. So with the hallucination, you are able to generate folds I have never seen in principle. But with the MP and N, you are able to design sequences that fit a given fold, right? So my question would be, if I have a protein family that they all share the same fold, more or less I can recover the sequence entropy that fits into that fold, but that follows an evolutionary trajectory. How much do you think your algorithm is able to design sequences that are unexplored evolutionary trajectories? Or what's your opinion about that? I mean, do you think really you are designing new things that have not been explored? Or
yeah, well, I think the sequences will be pretty similar. Like I showed you, the native sequence recovery is pretty high. So I think if you give it a native protein structure, it's going to come out with a sequence that is very similar. You know, like I said, 57% identical. So the method is actually too good at recreating native sequences to come up with new ones. Now, if you change the structure a bit, then you can come up with more new sequences.
Okay, thanks a lot.
That was a fascinating talk. Thank you. I have two questions. How do you deal with post-translational modification when it comes to in silico design and expression? And second question is there any, how does the field go about bioethics in this topic?
Right. So on the first one, post-translational modifications, you have to treat the first problem is in a prediction scenario, you don't know where they are a lot of the time. So it's a little bit easier in the designs case. So we have designed, for example, sensors of kinase activity where there's a serine residue that gets phosphorylated, and then we take that into account in the design calculations. And frequently, like in the design of, we've designed influenza binders, and there we have to take into account positions of the glycans. So we have to have additional information to model post-translational modification. As far as bioethics goes, important question. I think in terms of the potential of doing very bad things, I think the sequence of the 1918 Spanish flu is publicly available, and I think that's already probably much more dangerous than anything anyone will be able to design with protein design for quite some time. But I think it's an important thing to keep in mind.
Thank you.
Hello, this is Alex Iparola from Cincinnati Children's Hospital. Absolutely fantastic talk. Really enjoyed it. So I have two questions. One is about your first part about therapeutic applications, like picomolar inhibitors and these nanomolecule cargo carriers. So how do you deal with immunogenicity of those synthesized proteins, and do you have any factor in or objective function to make them more humanized, kind of reminding human-like sequences?
That's a really good question. So we haven't seen for these small. So first of all, for the vaccine I talked about at the beginning, it's obviously immunogenic. We want it to be immunogenic in that case. For these small binding proteins, we've looked pretty extensively for drug neutralizing antibodies and haven't observed them. So for example, you can repeatedly administer them over several weeks and they don't lose any efficacy. Administer them prior to challenge. So we have developed methods for eliminating obvious T cell epitopes, but the problem is that it's easier to do this in mice than in humans because humans are pretty polymorphic. We think the reason the small proteins are not very immunogenic is because they're very stable and very small and very soluble. And we have the first human clinical trials going on of a small design cancer therapeutic. So I guess the answer to your question is, so far it hasn't been a problem, but we're going to learn a lot more as more human trials happen. And there are computational approaches, as you suggested, to deal with the problem if it turns out to be important.
Mm-hmm. Thank you. And the second question is, you demonstrated those fancy designs like ladders, cages, and so on, so that assume lots of chemical interconnection. So how those proteins are synthesized, are they still utilizing ribosomal machinery? Because I would assume it's a linear synthesis, so how would you get these super interconnected molecules?
Yeah, everything I showed you today was made just using by ribosomal synthesis in cells. We have used to extend these methods to unnatural amino acids and unnatural backbones. And in that case, we have to chemically synthesize them. But what I talked about today was all just synthesized in cells.
Beautiful, thank you.
Hi, thank you for the talk. Your work is as impactful as it is cool in sci-fi. So one thing I noticed looking at the designs was a clear alpha helical bias. I know for the Rosetta designs, that's on purpose because a lot of this was built based off of alpha helical repeat scaffolds. But I'm wondering, is that bias sort of carrying over into the deep learning approaches? Is that consistent with what you're seeing? And if that's the case, how do you plan on breaking that, potentially branching out into more beta or coil structures?
Yeah, that's a good question. So there was a little bit, I think, of selection bias on my part. We have actually, it's true that we were able earlier on, and the field was able earlier on to design alpha helical proteins. But in the last few years, we've gotten better at designing all beta structures. For example, even the things as complex as transmembrane beta barrels. And so I just, for whatever reason, didn't select beta examples to show. And you're also right that the deep learning methods really don't have much of well, if you give them a short piece of helix, they will try and make helical bundles. But we also get all beta solutions when we do the inpainting, or largely beta inpainting and hallucination solutions. So I don't think there's any very strong bias. It will basically choose the secondary structure and topology based on what it thinks is best for scaffolding the motif.
Thank you. And my second question is, a lot of the structure-based approaches that you're describing usually require the construction of a multiple sequence alignment. Obviously, with design proteins, you can't easily get that because it's not native. So how do you circumnavigate that in your design protocols?
Well, yeah, that's a good point. So obviously, yeah, in design, you don't have a multiple sequence alignment. So, the networks You know, things like TR Rosetta, roseTTAfold, and AlphaFold don't work very well on native proteins when you just have a single sequence, but actually designed proteins are so the proteins we designed, we've kind of over engineered it. So they actually do quite well with single sequence information. So yeah, so everything I described today was just using single sequence information. We don't generate an MSA because, like you said, there really wouldn't be one.
Thank you.
Hi. Yeah, great talk. It's really exciting to see what you have going on. My question was about the process of going from your computational designs to your physically realized designs. Obviously, you could do it before with using Rosetta. And I was wondering if you're seeing like a quantitative difference in how quickly you can do that, and maybe how many iterations or how successfully you can do that as you're switching to these deep learning methods.
Yeah, well, let's see. There are a couple things that are happening. But yes, the deep learning methods are, you know, I guess it's a little bit hard to answer because, you know, when we pick any problem, you know, usually our success rate is really, really low. And then by the time we've been working on it for a while and really figured it out, our success rate can get quite high. So it's definitely true that with the deep learning methods, we are now getting higher success rates than we were earlier with Rosetta when we look at comparable problems. But we've also been studying them for longer. But I think it's also, I'm glad you brought that up because I should emphasize that, you know, what I showed you today were the designs that worked, but many designs still, when we have something that looks beautiful on the computer, but when we make a synthetic gene, put it into bacteria, you know, we get junk at the bottom of the tube. So design success rates are still far from 100%, but they are getting higher all the time.
Okay, thank you.
And let me just turn this back because we have a few questions from the audience live. So from Megana, are there applications from your work that can address problems in climate change, ranging from carbon capture, biofuels, and species conservation?
Yeah, that's a really important question. One of the issues, well, so yes, we're working on enzymes for you know, for things like bioremediation, and we have worked on new carbon fixation pathways because, as you know, Rubisco is not very efficient. The problem we've run into, though, is one of scale because, you know, to make a dent in something like you know, CO2, you know, in global warming, you really need very large scale. So what we're excited about now are sort of hybrid materials where you have an inorganic material which is most of the mass, but the protein is just a small part of it, but it's carrying out a key function. For example, catalysis. So we're trying to learn how to make those materials because there's a limit to how much protein you can ultimately make. I will say with those light harvesting systems, we are trying to design ones that would expand the light capture wavelength range of you know, the native photosynthesis, and we're hoping to put those back into organisms to see if we can actually expand that range. So it's a very important problem. The most obvious way to get to scale is genetically modified organisms, but as you know, those face challenges. So I think hybrid materials could be a way to address a lot of these challenges.
Fantastic, thank you. And another one from Vasilis. Have you ever tested if the hallucinated sequences might be encodable from uncharacterized genomic regions?
Oh, yeah, that's a good question. Well, it's certainly possible. We haven't looked, but, you know, sequence space is so large that it's pretty unlikely. You know, these proteins are around 100 residues, so the total number of sequences is 20 to the 100th. You know, it's like 10 to the 130th power. So the chance that we would generate something that was in a genome somewhere is pretty small. But I think it would be interesting to look.
Cool. And the last question we have online from Bryce: how does your work view the plethora of deep mutation scanning data sets and methods that have come out? Is this simply outside the scope of the work, or do you think it is not as helpful when designing these proteins far from wild type?
No, I actually think these methods generate really great data, and we are, you know, we look at them from time to time to evaluate our models. For example, our sequence design methods. So I think they're really valuable, and the more there are, the the more possible it will be to include them as actual training data.
Cool. And one from Sandun, is there a way to measure how accurate, reliable structure prediction is without comparing it with the experimental structure? Some users are predicting structures of random sequences.
Well, methods like AlphaFold and roseTTAfold will give you confidence measures, and but that's different from assessing reliability. So, I think when you use them outside the domain in which they were trained on, then there's always a risk, and that's sort of like what we ran into with the AlphaFold hallucination stuff, where we got sequences that AlphaFold loved, but they don't actually fold. So so you just have to be careful.
Fantastic. We have one more question from the audience.
Yes, so my question was about your hybrid materials approach. Have you thought about applying your peptides that interact with viral proteins to kind of maybe filter out viruses from the air?
Yeah, so in masks, yeah, we've thought about it in that application. We've also thought about, and we've done this with Theo at Tufts, who does amazing things with synthetic silk. He's actually made masks that have the sensor embedded in them, and they actually detect the virus in that context. So I think it's a really interesting idea. Again, there's an issue of scale a little bit, but that's certainly an interesting application.